Thank you very much for giving me the chance to present my research here. Within the next couple of minutes, I'd like to give you some insights into complex catalytic systems and how we can explore them using ultrafast, multidimensional, and computational spectroscopy. In this respect, I will mainly focus on metalloenzymes as catalytic model systems, in particular because they're major research targets of my work. And so throughout the talk, we will address the following questions. First of all, what are metalloenzymes and why are we interested? Secondly, how can we explore them? And last but not least, what can we do with the gained knowledge and the established methodology? Before we get to this point, let's shortly remember what we're actually talking about when we're dealing with catalysis. In the simplest case, we basically want to convert a certain molecule into another type of molecule via chemical reactions. And to do so, we have to get over this kind of energy barrier. The problem is, the higher this barrier, the slower a reaction will be, up to the point where it may not progress at all. However, what we can do is we can add a catalyst to the reaction. In this way, we will lower the barrier, thereby accelerating our reaction. This may still be a bit abstract, so let's shortly also remember what are the key features of chemical reactions by looking at this very simple example here. So the cleavage of molecular hydrogen into protons and electrons. And the first thing you can recognize is obviously that chemical reactions in the strict sense are always concerned with the making and breaking of bonds. And this is actually what gives rise to this barrier shown here, typically. Secondly, chemical reactions always release or consume energy. So in total, we can say that we are basically dealing with a transformation of matter and energy. And this is basically what we want to optimize in catalysis. Now, obviously, we are not the first ones interested in this aspect because since the emergence of life on Earth, every single living being had to deal with this task. And so it's probably not a surprise that nature has established some pretty sophisticated strategies for catalysis. And if we want to advance catalysis, it's probably a good idea to have a look how it's done in nature. Very briefly, in nature, biochemical reactions are catalyzed by enzymes. These are huge proteins, as you can see, consisting of thousands of atoms. So we typically represent them in this kind of schematic manner. And if we do that, you can also see that many of these enzymes contain catalytic metal ions, and so we call them metalloenzymes. These metalloenzymes have a couple of peculiar features. First of all, they catalyze their reactions with very high efficiency and specificity. Secondly, they do so by only utilizing earth-abundant and cheap base metals. And last but not least, they operate under mild conditions in a sustainable manner. So we could say metalloenzymes are just very great catalysts. But they are actually more than that. And to illustrate this, I want to refer to a model metalloenzyme called hydrogenase, which actually catalyzes the reaction we've just seen by using a sophisticated bimetallic active site. This alone is already pretty nice because, as many of you know, hydrogen is an ideally clean fuel which does release large amounts of energy upon combustion but no greenhouse gases. But actually, these enzymes also can do more. So for instance, many hydrogenases also catalyze secondary reactions, like for instance, the conversion of oxygen into water. And so many metalloenzymes in general are able to couple multiple catalytic reactions. Moreover, they are also able to synchronize these catalytic reactions with other non-catalytic reaction steps. And last but not least, we should be aware that the protein is not just some kind of bulk matrix, but it's a highly complex reactive environment. And so it's probably fair to say that metalloenzymes are not just simple catalysts, but they're actually complex catalytic systems. And that's why we're actually interested in the cluster of excellence unifying systems and catalysis. So the next question then is, if these metalloenzymes are such great catalysts, why don't we just use them as catalysts? Well, the point is that there are a couple of problems. First of all, these enzymes, as I just pointed out, are very large and very complex, and only fewer of their thousands of atoms are actually involved in the catalytic reactions we are interested in. And so what we actually would like to do is basically use them as an inspiration to design better, small synthetic catalysts that really fit our needs. However, this is also not really easy because, as I said, these systems are very complex. So in many cases, we don't really understand how they actually work. And so it's very hard to adapt their catalytic principles for human utilization. The key task then, of course, is we need to better understand how these metalloenzymes work. And the question is, how can we do this? Well, first of all, obviously, we need to understand the structural properties. But since we're dealing with catalysis, time is also a very important factor. And so the static structure alone is not sufficient, but we need to understand the dynamics of these systems. Moreover, we need to keep in mind that we're dealing with biological molecules, and so we need to make sure that we perform our experiments under conditions that are biologically relevant. How can we do this? How can we possibly literally shed light on metalloenzymes? Well, to answer this question, we may refer to another wise man's quotes here. It's from Richard Feynman, who said that everything that living things do can be understood in terms of jiggling and wiggling of atoms. And what he's referring to is basically the fact that molecules are not static entities, but their atoms are basically oscillating all the time, as you can see here in this animation. This looks pretty much random, but actually these motions are not random. They're just a complex superposition of what we call molecular vibrations, very well-defined atom motions, as shown down here. For instance, this oscillation of the carbon-oxygen bond at the active site of our model metalloenzyme hydrogenase. Now, talking about molecular vibrations, the question is, of course, first of all, why are we interested? Well, pretty simple, because these molecular vibrations depend on the structure of a molecule. So in turn, if we understand molecular vibrations, we can also understand the structure of molecules. Secondly, as we've just seen, these molecular vibrations basically constitute the dynamics of a molecule. And so the next question would, of course, be, how do we actually know that they look like this? Well, the simple answer is that we can calculate them using either classical or quantum mechanical methods. And this is one of the things we're doing in my group. The last important question is, of course, if these molecular vibrations are so informative in a structural and dynamical sense, then how can we measure, how can we experimentally detect them? And the simplest way to do this is basically by using infrared spectroscopy. Very simply speaking, this works as follows. Basically, you shine infrared light on your sample, in this case, our metalloenzyme, and if the frequency of this oscillating light wave matches the frequency of your molecular vibration, then you get a peak in your spectrum and you know, okay, I have this kind of vibration in my molecule, which also tells me something about the structure of my molecule. It's probably fair to say that infrared spectroscopy is not the only technique you can use to study molecular vibrations, but it has a big advantage over other possible techniques, and this is that it's 100% biocompatible, which is of course very important if we're studying metalloenzymes or biological targets in general. However, there's still a certain drawback with this or many spectroscopic methods, and this is the larger your systems get, the more complex your spectra become. And in the end, you end up with something like this, which looks not too different from the silhouette of a city, and it also has a similar problem like the silhouette of the city. Namely, you can extract some amount of information, but you definitely don't get a clear and complete picture. So how can we deal with this problem? Well, maybe we have to think about the way how we do infrared spectroscopy. Maybe we have to move on to advanced infrared spectroscopy techniques. And what you have to do to accomplish this is basically replacing your ordinary infrared light source, which is roughly speaking something like a light bulb, by a laser, which emits ultra-short pulses of infrared light. And if I say ultra-short, I mean about a millionth of a second, or even less. Now, if you use just a single pulse, then the result will be pretty much a normal infrared spectrum, so there's not much information that can be gained from this, or extra information. However, if you add a second pulse, then what you get is a so-called pump-probe spectrum. And as you can see, now we get an extra set of signals of opposite sign. So figuratively speaking, we're taking a look below the surface. This allegorically becomes even more clear if you add another third pulse, because then you end up with a so-called two-dimensional infrared spectrum. And as you can clearly see here, you have much more signals than in the normal infrared spectrum, and so you can also extract much more information about structure and dynamics. To illustrate this exemplarity, I next want to refer to or return to our model metalloenzyme hydrogenase. In particular, I'd like to refer to some proof of concept study that we just published last year together with collaboration partners from UniSysCat, the University of York, and the STFC Central Laser Facility in the United Kingdom. And what's very nice about hydrogenases from an infrared spectroscopic point of view is that they give rise to some very well-defined signals that basically reflect the stretching vibrations of these diatomic ligands here at the catalytic metal site. In the following, we will focus again on the stretching vibration of this carbon-oxygen bond, which we mentioned before. And if we recorded pump-probe spectrum of this particular vibration, then we end up with something looking like this. As you can see here, we have 4 signals instead of 1. And these 4 signals basically correspond to the excitation of different vibrational levels. What does that mean? Well, simply speaking, it means that the higher we climb this vibrational level, the stronger our bond will vibrate. And that's quite nice because from the energies we need to make it vibrate stronger, we can learn something about the properties of this bond. While I don't have the time to go into detail here, the essential take-home message is basically that we can determine the strength of this bond, and we can also determine the energy we would need to break it without actually breaking it. And this is of course quite neat, especially if you remember that the breaking of bonds and the energies associated with this process are 2 of the key features we are concerned with in catalysis or in chemical reactions in general. In addition, we can also determine bond lengths, and what I want to point out here is that these bonds are very short, so about 1 angstrom in this particular case, and this corresponds to about 1/10,000,000th of a millimeter, and we can determine these bond lengths with ultra-high accuracy. In principle, more accurate than any other experimental technique. And so together with the insights into bond strengths and dissociation energies, this approach, of course, provides very detailed insights into molecular bonding, for instance, into the catalytic function of this biological uncommon CO ligand here at the active site of our model metalloenzyme. So now that we have seen that we get structural insights, we also turn toward dynamics, because the signals here in our pump-probe spectrum, they are not static, but they're actually decaying as a function of time, as you can see here on the right-hand side. The reason for this is the following. Once we excited our molecular vibration, it will also get damped as a function of time, and so we basically return from these vibrational excited states back to the ground state. As you can see here on this plot, this happens on a very fast time scale. So again, this is picoseconds, so a millionth of a second. And what this tells us basically is that energy can be dissipated from the catalytic metal side towards the protein matrix extremely quickly. This is important for several reasons. Inter alia, it basically tells us that the general assumption that the rate of a chemical reaction can be related to the height of this energy barrier is correct, also for a catalytic metal site deeply buried within a protein. So now that we've seen some type of data we can extract from pump-probe spectroscopy, we may also refer to 2-dimensional infrared spectroscopy. What is important to remember here is that all these signals that we see will typically be projected into this plane here so that we can easily recognize this. And if we do this for this carbon-oxygen vibration we've just discussed before, then the spectrum will basically look like this. As you may have recognized, these signals here are basically identical to the one that we got from the pump-probe spectrum, so there's no additional information in this particular case, but it gives you an idea how the data look like. However, we can also extract additional data in other parts of the spectrum, much lower intensity, as you can see here, and there are 2 important things to consider here. First of all, these signals shown here on the left arise basically immediately after we excite the molecule, while these signals here grow into the spectrum as a function of time, approximately 45 picoseconds later. The second important aspect is that in both cases, the excitation frequency is the same, but the detection frequency in this case here is completely different. And what this tells us is basically that the energy that we've initially put into this carbon-oxygen bond, after about 45 picoseconds, is transferred to these carbon-nitrogen bonds shown here. And that's quite interesting for several reasons. First of all, because it tells us that 2-dimensional infrared spectroscopy can basically provide information about the flow of energy inside molecules. Secondly, we can also use this technique to study complex catalytic reaction mixtures containing different molecules or molecules in different states. The reason being that these kind of cross peaks we've just seen will only occur if the molecular groups, so in our case these 2 different types of bonds, are part of the same molecule. And last but not least, we can also extract additional structural information Inter alia, because again, these cross peaks will only occur if the molecular groups, in our case, these two different types of bonds, are part not only of the same molecule, but also if they are spatially close together. Needless to say, all this information is of course highly relevant to understand catalytic systems, like for instance metalloenzymes, especially for understanding the catalytic function and how this catalytic function is linked to other non-catalytic processes, like for instance the transfer of products and products. While all these techniques of course can provide a large number of other types of information, I would like to spend the last couple of minutes by giving you some insights into future perspectives, so what we want to do in the future. First, of course, we want to obtain additional insights into structure. First of all, experimentally, by not only probing these catalytic metal sites itself, but also their protein environment, which, as I mentioned, is highly important to understand the catalytic function. Moreover, we also want to complement our experiments by additional computational studies in order to simulate data from advanced infrared spectroscopy, thereby facilitating their understanding. Then, besides static structure information, we also want to obtain more dynamic information. First of all, by exploring how pump probe and two-dimensional infrared spectra change throughout catalytic reactions, which will tell us something about the catalytic mechanism and the involved intermediates. And secondly, also by exploring equilibrium dynamics for each of the involved intermediates, which will, on the one hand, give us further mechanistic information. It may also give some insight into the way how energy and information is transferred within large molecules or whole catalytic reaction networks. Last but not least, we also want to further improve in terms of biologically relevant conditions, and so we also aim to record advanced infrared spectra from metalloenzymes within whole living cells, which is basically an expansion of a previously established in vivo spectroscopic approach. Of course, we don't want to use these techniques only to study metalloenzymes, but we also want to use them to basically investigate other catalytic systems that are relevant for the cluster of excellence UniSysCat. And last but not least, we also aim for a change of paradigm. And what I mean with this is the following. Typically, when we're doing spectroscopy, we're basically dealing with the interaction of light and matter. And usually we use this interaction to understand the structure and dynamics of molecules, like for instance, catalytic systems. Once we gained this understanding, however, it would also be nice if we could use light also to control or maybe fuel catalytic systems. And this basically brings me back to the title of my talk and the original motivation. And with this, I'd like to thank you for your interest in my research.